Today we're going to learn about some of genome-wide techniques, which goes under the umbrella of omics. So basically, here's some terminology that we use. We already seen some of this. So the study of the genome structure, function, and evolution as a whole is called genomics. And we talked about the RNA and the kinds and types and the amounts of RNA in cells and organisms. We call that the transcriptome of the cell or the organism. And the study of this is called transcriptomics. And likewise, for the protein, the types and kinds of proteins in a given cell or the organism is called the proteome of that cell or organism. And the study of this is called proteomics. Okay, so today we're going to talk about a little bit about proteomics. We're going to start with that because you've already learned quite a bit about genome structure and function and genomics and transcriptomics in the past few lectures. All right, so we learned lots of techniques about how to analyze the amount of DNA, RNA, DNA sequence, RNA sequence. And you already learned a bit with Professor Siegel about some of the techniques that you can use to analyze proteins, right? So typically identifying and quantifying proteins in the cells are done through techniques such as centrifugation or column-based separation techniques based on the property of proteins. Different proteins have different properties such as the length of the protein, the charge of the protein, and the shape of the protein, etc. So these methods, the principle of these are based on separation of the proteins based on their different different properties, right? So one of these is gel electrophoresis, and you learned that the smaller proteins run faster and the larger proteins run slower, and that basically separates them. Okay, so that is usually one-dimensional gel electrophoresis. That's that you separate the proteins based on just one property. Okay, so there's also other techniques such as two-dimensional protein separation. So in this technique, you separate the proteins based on two different properties. So here's how this is done. In the first dimension, what you do is you put your protein mixture here above a matrix, like a gel matrix or whatever property that you want to separate them with. And so in this gel, you are separating the proteins by charge first. Okay, so your gel separates proteins such that the proteins with more acidic proteins do not run as fast as some of the more basic proteins. So this is the first dimension of separation by charge. And then what you do is you take this little gel piece and you put it on top of another gel, okay? Now horizontally, so that all your proteins that were separated that are acidic are on the left hand of this little gel strip, and all the proteins that are basic are on the right hand of this gel strip. And now you put this gel strip on top of this second gel, and now what you do is you separate them by size, okay? So the properties of this gel and the buffers that you run in this run these proteins within the electrical field allows you to separate them by size. So that's essentially what's SDS electrophoresis. So what you have is that if your protein is acidic and large, it's going to be stuck in the upper left corner of this second gel. And if your protein is basic and small, it's going to be running fast, and it's going to be on the right hand of the gel. Okay? Does that make sense? All right. Yeah?
What does SDS stand for?
Sodium dodecyl sulfate. Yeah. All right. So And then what you do is you can label these proteins somehow to visualize them, and some of these labels could be radioactive or some other type of labels, right? And in this example, the proteins are radioactively labeled, and a typical result will look like this. So every dot here is going to be a one protein that is separated by size and separated by isoelectric focusing, basically the charge of the protein. All right, so what can we do with this kind of stuff, right? What can we learn from 2D gel, two-dimensional gel? That's what we call them. So you can compare the level of a protein between two samples. So if you're interested in what proteins go up in a cancer situation compares to wild type, or some other situation like the muscle cells versus nerve cells, and you want to know the protein amounts, you can compare that. And so how do we do this? Essentially, you can do a 2D gel proteome map. And in this example, we're looking at a 2D gel proteome map of prostate tissue from tumor versus benign samples, normal samples. And what you do is you isolate from proteins from normal and cancer tissues. And then in each sample, you conjugate proteins to either CY3 or CY5. So what you do is you take a fluorescent molecule like CY3 and CY5, and you chemically link that to every peptide in your mixture, right? Every protein in your mixture. And then you you mix them together, and then you run your 2D gel, okay? And if one tissue has more of one protein, then the spot will be more of the color that you label the proteins with, right, in that sample. So for example, here, if we labeled the cancer sample with CY5, CY5 is red, right, and I look at this spot here, there is a protein here that appears more red than green, what would you say about this protein's level between the cancer and the normal sample? Cancer sample is labeled by red, and you're seeing a spot here that's more red than green. What is that?
It's a protein that's more specific to cancer.
Yes, it's a protein that's more expressed in cancer tissue that you looked at, right? What about this protein here? So there's this little yellow spot. It's expressed in similar levels in both samples, right? Yeah, this is essentially looks like a microarray, right? So what differential analysis technique does this resemble? It resembles microarrays because you're labeling one sample with one color and the other sample with other color. You're mixing it up, and then based on the differential amount of the color, you infer that the amount of the protein is different, right? What's one of the biggest difference from microarrays, though? So when you look at this spot here, in microarrays, if you look at a spot, do you know what gene is that? Yes, because you printed that, right? You know that every probe has a specific sequence, and you know that sequence because you printed that little probe on the glass slide. Do you know the identity of this protein here? No, you don't. You know that it's a protein, it's a specific protein because it's separated from everybody else, but you don't know what the identity of that protein is, right? So then, if you want to couple your 2D analysis with the analysis of a proteome, that means you want to know the identity of every protein, you need a technique that will be able to tell you what protein runs at this location in a 2D gel, right? So you want to know what protein is at that each of that spot. And people have done these kind of experiments. For example, this is all the proteins that are in a heart tissue. And then what they wanted to do is sequence all of these proteins and find the identity of all of these proteins. Sequencing nucleic acids is easy. We have lots of techniques to do that, okay? But protein sequencing is a whole different matter. It's much more challenging. So, and there aren't that many techniques. But one of the techniques that actually allowed the explosion of the proteome field is mass spectrometry. Okay, so this is a technique that will help you identify and quantify proteins. So some of the questions that it will answer is how to determine the sequence of many proteins in a sample. Basically, you can use mass spectrometry. It's an analytical tool that's used for measuring the molecular mass of a peptide fragment, and the molecular mass of a peptide fragment can tell you something about the identity of that peptide. And combined with the genome sequence, that is, you know the coding potential of your genome, what proteins can your genome actually encode for, you can combine these two information and infer the proteome of your sample. All right, so let's explain this a little bit. What is mass spectrometry? It uses an instrument called mass spectrometers, and these are made up of 3 components. One is an ionization source. So what it does is it takes peptides, right? Peptides is long chain of amino acids. And then it transfers, so you prepare these peptides so that these peptides don't have charges normally. And then you use this ionization source to transfer a charge, a proton, to the peptide. So the charged peptide is now called an ion because it's a charged molecule. Okay? The second component is a mass analyzer. What it does is it separates these ions, essentially the charged peptides, by mass to charge ratios. Okay? So since most of our peptides will have the same charge, not always, but most of the time, the mass analyzer is going to be separating these by their mass. Okay? And the mass analyzer does that by using electromagnetic field. And the third component is called an ion detector. Okay, so these ions are charged, they're separated, and then the detector takes these separated ions, which is a strike to itself, then measures the ion current over time, and the more current, then it's going to indicate the amount of signal at each m to z value. Okay, so how does this work actually? So here's one example of there are different types of mass spectrometers. Okay, so this is one type. It's called molecular matrix assisted laser desorption ionization time of flight. You don't have to remember this, but multi-top mass spectrometry. The principle is this. The separation is based on the mass of the proteins and the fact that when proteins are running across a magnetic field, the smaller proteins run faster than the larger proteins. Okay, so that's basically essentially the separation principle. So what you do in this case is you have a little metal plate where you place your sample. So your sample is a mixture of proteins or peptides, right? And then you use a laser to ionize the peptides on this plate. And then you use a magnetic field to accelerate these peptides from the plate to the detector, right? So these peptides are going to strike this detector. And the field is such that the smaller peptides are going to fly faster. They're going to run through this faster, which is called time of flight, right? So the time of flight is going to be disproportional to mass. The larger you are, it's going to take longer for you to fly, right? So the lightest ions are going to arrive at the detector first. And depending on how many of these small ions or large ions is, the detector is going to measure the charge. So if there's a lot of them, it's gonna say at this mass value, I'm seeing this much of charge. So it's a lot, and then at this mass value, I see a little less. At this mass value, I see a little less, etc. Okay, we're gonna see how we actually express that data in a bit. All right, a second type of mass spectrometer is called ion trap mass spectrometry. So the principle of this is a little bit different in that the mass analyzer does the separation here using electromagnetic fields. So you're not, the ions separation is not based on time of flight. Okay. So in this case, what you're doing is that you're taking your sample, which is in liquid, it's a mixture of peptides and proteins, right? And then you put it into a needle, which is an electrospray needle. And this needle shots the sample into an electric field, okay? So this electrospraying ionizes the proteins, ionizes the peptides. And each of these peptides basically form droplets of these ions. And then this is released into a detector, then which detects how much of an ion with a certain mass is present. Okay, so this mass analyzer separates the ions based on mass and strikes it into a detector, and the detector is going to detect the amount of charge coming from a given mass value. So how do you express that data? So you express this mass spectrometer data using what we call mass spectrum. Okay? So the detector is measuring the amount of charge for a given mass. Okay? So what you do is you basically put the mass value in the x-axis, and then you in the y-axis you plot the relative abundance of ions that's coming from the detector, telling you how much charge it's seeing. So, for example, in this example, there is in your sample there was a peptide or a protein of 565 mass value that was very highly abundant. And then you had some other peptides, for example, at 700 mass value that was less abundant, right? Does that make sense? All right. So the x-axis of a mass spectrum represents a relationship between mass of a given ion and the number of elementary charges it carries. It's usually just 1. So the mz value basically is going to be a number proportional to mass. So in the x-axis, it's essentially the mass. In the y-axis is the relative abundance of the ion. So each of your samples is going to be composed of different proteins and peptides of different masses, right? So if you run your samples through mass spectrometer coming from your heart sample, the spectrum is going to look different than samples coming from your nerve sample, right? So if you're comparing the proteins in two samples. So they're going to have a different mass spectrum. And this is typically used for when people want to know, for example, they pick up a sample at a site and they say, is this sample same as some other sample that I know? Okay, is this have the same composition? And you can just simply compare the mass spectrum and say it's the same or not. Okay, but this by itself is not telling you the protein sequences, right? So you have to go one step further. These type of samples, complex samples, contain large peptides and large proteins, and sequencing of these large peptides and proteins is impractical, okay? So what people do is then they take these samples, right? So they have the mass spectrometers are set up in a way that they will take the sample at this mass ratio, okay, at this mass value, and then they will break that peptide into smaller peptides, okay? So you take that peptide and break it into smaller peptides and do the mass spec again. So this is tandem mass spec. You do one and then you do a second one. So when you do the mass spec again, you're taking, let's say, a 100 amino acid protein, and then you break it up into 25 random smaller peptides, and then you pass these mass spec again, and each of these smaller peptides is going to give you a different mass spectrum. Right? the mass spectrum for smaller peptides is going to look something like this. Okay, so this is the mass spectrum from a peptide of this sequence is shown here. So this is the amino acid sequence of this peptide, and then so for this peptide, depending on how randomly it's actually broken up, you're going to generate this many possible peptides from this, right? And then each of these peptide masses is going to give you a relative abundance. So from a peptide sequence of like this, by randomly breaking into smaller pieces, you can get this spectra. Okay, so this spectra is basically coming from a mixture of peptides, okay, that are produced from this small peptide. Okay, so all right, so each short peptide will have a different mass spectrum. So basically, that's what we call a mass fingerprint. And then what you have to do is you have to take this mass fingerprint and then match it to a database that contains all the possible mass spectra coming from all the possible amino acid sequences of short peptides. Does that make sense? So each of these short peptides is going to have a stereotypical mass fingerprint. Okay? Once you make that mass fingerprint, you can go back to the database and ask the database which of the amino acids is actually giving me this mass signature, and then once you have that signature and you have the sequence of the protein, that short peptide that you have. All right, so a summary of this analysis is basically you have a complex mixture of proteins. You break these proteins into peptides and then separate them. And then you can take each of these peptides and break them into smaller peptides so that you can get a mass fingerprint for each short peptide. And then you can take these mass fingerprints and match them to a database to tell you which protein sequence, which amino acid sequence can produce this mass fingerprint, okay? And then now you found the identity of a short amino acid region of your original protein, right? Yep.
So when it goes into the mass spec at first, you haven't natured it or done anything to it. It's just a perfectly folded protein, and then it's degraded into random peptides.
It's a little more complicated than that. Usually it's not full proteins. You first break them using a protease into smaller pieces because a lot of the proteins are too big for the mass spectrometer to handle. So you take your proteins, you break it into peptides. These are long peptides. And then they go into the mass spec so they get separated. And then these long peptides get broken into smaller pieces that produce the little mass fingerprints that you use to sequence them. You guess the sequence of what these peptides are. Does that make sense, guys? All right? Okay. So basically then you get short little reads, right? You get a short little stretch of amino acid reads that will tell you that's the sequence of a protein in your sample. That's what it says. Okay, so that a protein of that sequence is in your sample, and that read is short. It's not the whole protein. It's just a piece of the protein. Yep.
So a single protein is degraded, and that's what goes in.
Yeah.
Okay. So if it's a single protein, if we're comparing between two different tissues, like nervous tissue and heart tissue, are you just seeing if the protein shows up at all?
Exactly. Yep. So that's essentially what. So you're asking the quantification, and in this case, quantification comes in here. Okay. So the number of times you detect a certain protein in a sample, right, will be proportional to the amount of that sample in that amount of that protein in that sample. So if you detect a certain protein many times, right, and you take different regions of the protein, right, so that means that protein is abundant in your sample. And if you don't detect that protein at all, or you detect only one peptide from it, right, and only one time, not like 10 or 20 times, then you know the amount of that protein is less. Yes?
So mass spec is done with only one protein, like a single protein?
No, it's a mixture of proteins.
Oh, okay.
Right? But how do you know which protein? Because you know the sequence, right? Let's say it's a lysine, proline, histidine, blah, 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 right? You take that sequence and you go to your genome and say, which protein matches to this sequence, right? And you take that sequence and match it to all the coding sequences and say, this little peptide sequence is coming from RNA polymerase because I know that RNA polymerase has that sequence. Does that make sense? All right. So that's essentially comes to my question, is that what step does that resemble? You have a short peptide sequence, and then you match it to all of the possible peptide sequencing coming from the genome. What step does that resemble in ChIP-seq or RNA-seq?
Matching the reads back to the genome.
Very good. Matching the reads back to the genome. You know how we talked about annealing the reads based on the sequence? Right now, what you're doing is you're taking the peptide sequence instead of DNA or RNA and matching it back to the genome and finding out which protein and which region of the protein does that match. Does that make sense? All right, good. So proteomics is not the only not the only omics technique that's left here. Okay, so we have genomics, transcriptomics, and proteomics. Because our bodies are made up of DNA, RNA, and protein, but also other things, right? And some of these things are metabolites. So these are small molecules like hormones and sugars and etc. that our bodies use. And the other one is the microbes. So the human body is not just made up of human cells. We also contain a lot of microbe cells, right? We contain a lot of bacteria in our body. All right, so the study of all the metabolites in our body is then called, is the types and the kinds of metabolites in our body is called the metabolome, okay? And the study of is metabolomics. And all the types of and the kinds of bacteria in our body is called the microbiome, and the study of that is called microbiomics. All right. So the idea of our bodies having metabolites is really old. So, and the analysis of metabolites are also kind of old. Okay. So, for example, it was many hundreds of years ago that the diabetes was actually diagnosed by essentially the sweetness of urine, right? So in 1674, this English physician, Tom Tallis, was the first in modern medical literature to observe this relationship. And he may have enjoyed the sampling process a little too much, stating that the pea on his palate was wonderfully sweet, as if it were imbued with honey or sugar. So essentially, he tasted to diagnose. Of course, this is modern life. We don't want to taste people's peas, right? So there are other ways to actually measure metabolites in the body. So we basically determine the metabolic profiles with other methods, and these methods are nuclear magnetic resonance, NMR spectroscopy, and mass spectrometry. So these metabolites are small molecules. So these mass spec and NMR techniques actually can determine the chemical composition of these things. And I'm not going to go into that because it's a lot of chemistry. Okay? All right, so what do we do with this knowledge? So the human metabolome database is basically a database that was created in 2008. So it contains three types of data. So it contains chemical, clinical, and molecular biology and biochemistry data of about 41,000 metabolite entries, including water-soluble, lipid-soluble, abundant, or rare metabolites in our genome. Okay, so that was in 2008. In 2016, we see about 100 and actually this is 2017. I just checked it last week. There was about 114,000 entries. So we have a lot of metabolites, a lot of different kind of metabolites. But what we don't know is, now this is a catalog, we don't know how each of these are actually regulated and what their functions are. So what I mean by metabolite, right? And the most famous metabolome pathway, so it's the pathway is glycolysis, right? How we use sugar. So you can see a little bit of a diagram of this pathway. And each of these little chemical molecule that are shown here is a metabolite that's a product of this pathway. Okay? And the way that these metabolites are linked to this pathway is through these enzymes. So this, for example, this enzyme takes this metabolite and then produces this metabolite, right? So that's how the enzymes are linked to each of the metabolites. So the amounts and kinds of these metabolites is basically telling you how this pathway works, right? So if, for example, there is a condition where this enzyme is no longer functioning, then in that sample you would no longer see this metabolite, right? So it's the amount of metabolites is going to tell you something about how well this glycolysis pathway is working, right? All right, so this is just one metabolite from the most famous pathway. There are other metabolites that the human metabolome database contains. Some of these are basically drugs or drug metabolites, so similar to drugs. So there are about 3,000 something common toxins and environmental pollutants, and a bunch of pathway diagrams so that are linked to metabolomic diseases. And 28,000 food components and food additives. Yeah?
I'm sorry, what exactly are the drug metabolites? Are they like metabolites in the body?
So you take a drug and then your body metabolizes it, right? And then you create different kinds of little chemicals that would be drug metabolites, right? And the environmental pollutant, we actually saw some of these in the DNA repair lecture. So what are some of the environmental pollutants that can be metabolized by our body? Does anyone remember? Yeah. Tobacco. What else? There were some liver toxins, right? That was our body metabolizes and then creates some of these cancerogenic molecules that can damage our DNA, right? All right. What about microbiomics? So this is also mainly studied by many labs around the world, and also under a consortium project called Human Microbiome Project. Okay. So one of the first publications that came out of the Human Microbiome Project is, again, a cataloging type of effort, right? Let's sequence and find out what are the bacteria that live in our body, right? So they picked, so just to tell you how many bacteria and microbes live in our body, it's actually pretty substantial. So in an average human, the number of human cells is 10 to the 13, and the estimated number of microbes that live in the human body is 10 to the 14. So essentially, although we're by mass more human, we actually by number we're more bacteria, right? So how does that actually relate to mass? So for a 150-pound human, that's about 1.5 to 4.5 pounds of microbes, right? So you can think of it like a home for the microbes. So in this study, what they did is they took 242 healthy U.S. volunteers, this many male, this many female, collected tissues from 15 body sites from men and 18 body sites from women. And then they sequenced the DNA from 5,000 samples and identified the bacterial genus, right? So based on 16S rRNA, okay? And then what they found is they found there were more than 10,000 different species of bacteria that live in our body, okay? So some of the outcomes of this work was published here, essentially revealing what was found. This is probably not surprise to anybody that the different types of microbes are different types of body parts. So what's this plot here? So what this plot is doing is it's taking all the different types of microbes, the amount of these microbes, and separating them based on how similar they are. Okay, so they have 5,000 samples. Some of these samples are taken from the tongue. Some of them are taken from the skin, etc., right? And so they say, as all these 5,000 samples, which of these samples resemble each other the most? Okay, that's what essentially this is doing. It's, again, kind of a clustering algorithm, right? So it's looking at each of these samples and saying, how similar are these together? And if they're similar together, the plot is in a two-dimensional it's a two-dimensional plot, putting these two samples near each other, right? So if two samples are similar, they're near each other. And the algorithm basically colors each of these samples with a different color from where they are taken from, right? So all the samples coming from oral samples are basically clustering together. What this means is that for each of these samples that are taken from the mouth, the types and the kinds of bacteria are similar to each other, right? And different from the samples that are taken from gastrointestinal places, right? And those are different from the bacteria, etc., taken from the skin. Does that make sense? All right. And actually, I think more of the surprise was the fact that the microbial carriage, that means the types and amounts of microbes that each person carries, is very different, okay? So in this plot, what they're plotting is that they're plotting the relative abundance of this particular species. This is Streptococcus, right? Species in the tongue samples. So 127 tongue samples. And so each sample is shown in the x-axis, and the percentage of strep is shown in the y-axis. What you can see here, and the different species of strep is color-coded here, so that, for example, you know that this species of strep is this light blue right here, right? So you know that, okay, this species of strep here, which is this one, is very abundant in this sample, right? And the red Thermophilus strain is a little less abundant, etc., etc. So you're looking at the relative abundance of each strep species in the sample, right? And what you can see is that in this person, which has a lot of strep bacteria in their tongue samples, because 60%, more than 60% of all the bacterial species that came out of that sample was a strep species, okay? And within this strep, there are the individual species distribution are shown here. And then this person here has very few strep in their sample, right? And then the distribution of the different strep species within the sample is different from this. So essentially, this is summarizing the fact that if I went around here and took tongue samples from all of your samples will be very, very different. Some of you will have certain bacteria a lot more. Some of you will have certain bacteria a lot less. And the species within each group of bacteria, the distribution is also going to vary. Okay? So different microbe populations from different people. What could be some of the basis for this variation? These are all healthy, by the way. None of these people have strep throat. Diet, right? So environmental factors would be essentially the biggest contribution of this. All right, so what is this microbiome, right? You probably heard some popular science stories about this, all the excitement around it. What's up with that, right? It turns out the microbes that live on our body actually affect our health very much and is linked to diseases that we actually even did not realize that it was linked to. Okay? For example, in this very recent publication, the authors were looking at a colon cancer mouse model, and then what they found is that this treatment drug called gemcitabine that is usually used for this colon cancer was actually induced in some cancer mouse models was completely inactive. So basically there was gemcitabine resistance. And they were trying to figure out why this was, because there's humans with this resistance as well. So they wanted to figure out where does this resistance come from. And they could not find any genetic cause, but what they could find was that they realized that in these tumors there was some bacteria of this specific type of bacteria called gamma proteobacteria. And the correlation was that if that bacteria was expressing the cytidine deaminase, then that made these tumors resistant to gemcitabine. Okay? And they wondered, okay, this is really interesting. We wonder if it's actually also related to some of the human gemcitabine resistance. So they said, can we actually see this bacteria in the tumors that we isolate from people, right? And so this is essentially a figure showing you that. This is a pancreatic tumor. And what they're showing you is that they're doing a DNA fish against the bacterial rDNA from this gamma proteobacteria group, okay? And all of these little red lines here show that this bacterial DNA exists in that sample. And here's another type of assay in which they are trying to see if this bacteria is present in the samples. So they're doing an immunofluorescence to a bacterial protein. So this antibody only binds to this bacterial protein, and you can see the presence of this bacterial protein in this histological sample shown with these arrows, right? So this bacteria is also present in the humans. So then you can say as a doctor, so if I'm going to give gemcitabine to these people who might be resistant, maybe I will give them first antibiotic, clear all the bacteria, and then give this antibiotic, right? And this drug, and that makes sense. But that's not a approach that you can use widely. What that means is that in this case, the bacteria is not beneficial, right? It's not good. It confers resistance to drugs, but there are other cases in which specific bacteria actually are beneficial. So what's an example for that? So this is also another recent study where they are looking at the effect of gut microbiome in immunotherapy efficiency. Okay, so a very promising type of immunotherapy is the use of PD-1 inhibitors. The reason for that is PD-1 is a protein that enables the cancer cells to shut down the T cells. Okay, so the cancer cells evade the immune system by upregulating this PD-1 protein. So if you actually inhibit that, so you use PD-1 inhibitors, then the immune cells can actually attack the cancer cells and help clean up the cancer from the body, right? The problem is if it works really well, but if it doesn't work, it doesn't work at all. So they're effective, but only 25% of the patients actually respond to these PD-1 blockers. So what they found is that if you actually there are a bunch of beneficial bacteria that they suspect in the gut that increases the efficiency of these PD-1 inhibitors. So if, for example, the patient takes an antibiotic before the treatment, that actually reduces their chance of actually responding to the treatment, right? So that means that not all bacteria are bad. Some of them are good and some of them are bad, right? So, and for each kind of treatment, for each person, then we should know which bacteria are good and whether that patient needs to have an antibiotic treatment or not, right? So that's pretty cool, and that's what all that jazz is about, right? People are excited. All right, so now we know we can analyze the genome, transcriptome, proteome, metabolome, and microbiome, and it tells us something to do with the disease state and the normal state and normal development of our bodies. So then in principle, right, in theory, I should be able to compare the genome, etc., of two groups, right? For example, with or without disease, and then find out what distinguishes these two groups, right? And then that can serve as a starting point to test if these differences are causal, right? This would provide correlation, what distinguishes it, and then you can use that to see if it's actually causing the disease or not. So in theory, I should be able to sequence everyone here, right, and find out our DNA sequence differences, and then that underlie the genetic differences in the phenotypes. But of course, this requires knowing all the ways our genome varies and functions, which we don't know yet, but that doesn't stop us to move on to personal genomics and try to use that, okay? So because we know that genotype determines a large proportion of our phenotypes, right? So this genotype-phenotype difference. All right, so then we need to start with how much variation is in the genome sequence of two individuals, right? How different and how similar are we? So we're very similar, right? So on average, two random people share the same DNA sequence in 99.9% of our genome, so we're all human. But we're also very different. So given 3 billion bases in the genome, on average, two random people will then differ at about 3 million base pairs locations, right? So in 1 to 1,000 nucleotides. So that's very different. So just to give you an idea of how similar we are, we're 99.9%. The estimated sequence similarity between humans and the chimps is about 98%, right? So we're very similar to each other and different from chimps, but we're also pretty similar to that. But essentially, we know that the genome sequence determines the phenotype largely. So the phenotypic difference between this short person and the tall person basically lies in the genotype sequence. Then in principle, I should be able to sequence the short person and the tall person and find out what the differences are and if that causes how tall they are going to be. But it's not that easy. Okay, let's first talk about the types of differences between our genome sequence. We call these DNA polymorphisms. Poly means many, morphisms means form. So DNA polymorphisms, this can come in different flavors. One is called single nucleotide polymorphism, so the difference at one nucleotide locus, right? There could be differences in simple sequence repeats, microsatellites, so you can have the number of the different microsatellites. And you have differences in copy number of larger chromosomal regions that can change from 1 kb to several megabases, and this copy number variations has been really found recently to be linked to disease, these CNVs, okay? And these single nucleotide polymorphisms, which we're going to talk about, occur at a single location and doesn't affect large regions. Okay, so we want to understand phenotype to genotype relationship. So we need to learn a few terminology to understand this, okay? The first one is single nucleotide polymorphism, SNPs. You already learned a bit about this. So they can be sequence difference. They could be small deletions, like we call it indel, so inside deletion, just one nucleotide, etc. And these result from replication errors and unrepaired DNA damage. And if they are inherited through the germline, it's going to pass to the next generation, right? And that's all these mutations basically create this variation in our genomes. So there's about 1 to 1,000 SNPs on average between two individuals. And SNPs mostly occur in introns and non-coding DNA. So 2/3 of these occur in introns and non-coding DNAs, okay? The coding regions are mostly more similar than non-coding regions. All right. So a lot of studies that compare the two DNA sequence of two individuals don't sequence the whole genome. What they do is they sequence certain locations only, certain SNPs at certain locations, because we know that a SNP at a location is linked to other SNPs in the neighboring location. Okay, what does that mean? So groups of SNPs that are separated by recombination hotspots segregate together within the genome. What do I mean by that is that, so here's your chromosome. Okay, here's your gene X, and in this region, let's say there are 5 SNP sites, A, B, C, D, and E. And each of these sites, you have some variation within the population. And here are some other SNPs outside this region, X and Y, outside this hotspot. So during meiosis, when the chromosomes are exchanged, right, chromosomes get shuffled up, that exchanging occurs through DNA strand breaks, okay? If these hotspots are the locations where most of the a lot of DNA strand breaks occur. And because of this, okay, the sequences here don't get shuffled very much because they don't get crossed over, okay? So every time there's a meiosis occur, this little block here gets segregated to one chromosome or not. So the sequence here at A location, B location, C location, D location stays similar, okay? So they're just segregated by blocks. So because there is not much recombination between these SNPs, these A, B, C, D, E, and E SNPs, they are passed from parents to children as a unit, which we call the linkage disequilibrium block. Okay? So, and the sequences at these SNPs, A, B, C, D, make up a haplotype. There is a great study question at the end of this lecture. If you do it, you should do it. Do this study question, and then it's going to make it much more clarify to you what is haplotype is, because you're going to try and find it, okay? All right. So if a gene X causes a particular trait or disease, then a certain haplotype, so that's basically a particular sequence at A, B, C, D, E, will correlate with that disease. So what that means is that, for example, if there is a mutation of gene X here, that sequence is always going to be linked to the particular sequences that are A, B, C, D, E SNPs that's going to correlate with this disease. And that's basically the basis for genome-wide association studies. These studies try to find SNPs that correlate with disease, okay? They don't essentially know if a certain gene is correlated with disease, but they will be able to tell if a certain SNP is seen in a disease. All right, so how are these genome-wide association studies are done? They're basically trying to determine the allele frequency in two different groups that they're comparing, okay? So let's say I took the population in this room and sequenced your DNA and determined the SNPs in these groups, and separated you guys by students who love MCB and the students who meh MCB, right? And then I will have certain individuals, 1, 2, 3, blah, 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 and then some other ones in this group, 1, 2, 3, blah, blah, blah. And for each person, I'm going to have the sequence DNA sequence of each person, okay? And at some locations, you're going to vary. So these locations, for example, SNP1, SNP2, and SNP3, and the sequences that vary, I kind of color-coded in red, okay? So what we need to know is which of these sequences up here may be conferring love for MCB comparing to the other students, right? And right away, one of the things that you might be thinking is that we need to know what the normal variation is, right, to link a certain SNP to a certain phenotype. And we need so we need to know what's just normal variation and what is actually correlation, right? Because of that, you need to know the normal variation. You have to look at a very large population, right? So you need to examine many hundreds and thousands of people to figure out, for example, if the C is really correlated with people who love MCB, or it correlates with a whole bunch of other things, or that it's actually present in lots of people who may MCB, but you actually just didn't sequence enough, right? So you need to distinguish that. So these are the type of things that the GWAS studies do. They take lots of individuals and determine the SNP sequences within these individuals. For example, this is a study that took about 9,000 individuals, and they genotyped them not for the whole genome, they genotyped them for a small number of SNPs because it's expensive to sequence the whole genome of everybody, okay? So you pick certain SNPs within, across the genome, and because each of these SNPs are going to be linked to each other, so you can choose a smaller number of SNPs. So they looked, for example, for 22 common traits like hair curling, freckling, eye color, etc. And then what they could find was they could find some SNPs and genes associated with some traits. So how do we actually express this data? We use something called Manhattan plot because it looks like large blocks, right? Okay, so what's a Manhattan plot? So what you do is on the x-axis, you have each of the chromosome, and each of these dots is a SNP location, okay? On the y-axis, you plot the statistic test outcome telling you what are the chances of the SNP being correlated to the phenotype of your interest, okay? So if the SNP is correlated to your interest, it will have a low p-value, a high significance, right? And they're going to appear like this. So here, what you're looking at is that, so for photic sneeze reflex phenotype, there is a region on chromosome 2 that has a bunch of SNPs that's correlated with this phenotype. It's not telling you just yet what gene is correlated with this phenotype, but you can suspect that if a bunch of SNPs is correlating with this phenotype, there must be a gene at this locus close to these SNPs, right? Because these SNPs travel together, right? So there must be a gene that's close to these SNPs that's linked to this phenotype. So what then you can do is you can go to this region, find out what are these genes, and then study the function of these genes in more detail, whether it's correlated or not. All right. So these type of studies have really increased lately. A lot of people are trying to link the genotype to phenotype, so they do genome-wide association studies. So this is increasing. What are some of the outcomes, right? So one of the outcomes is that the fact that we actually don't understand the human. Yeah, go ahead.
I just had a quick question, because you said they typically don't sequence the entire genome, but in order to find the SNPs, wouldn't you need to sequence the genome?
Okay, so what they don't sequence the entire genome, but that they determine the sequence of specific SNPs. Okay. What do I mean by that? So let's say this type of linkage disequilibrium blocks are really large, right? So these are several hundred kbs. So you have two options. One option is that you sequence this entire region region, okay? Let's say that's 100 kb. I'm just making it up. 100 kb region you need a sequence, okay? But not each of these region is going to contain a SNP, right? So there are some of these regions that have coding sequences. They are going to have exactly the same sequence in between individuals. Then it's a waste of money, right? So you basically don't need to do that. What you can do is you can do an initial survey of the population and find out some of the spots that you frequently see SNPs, okay? And then you focus only on those SNPs, okay? Let's say in this example, instead of sequencing the 100 kb, what you need is you need a method, either microarray or sequencing, that's going to sequence these 5, 6, 7 spots, right? So you already don't have to do the whole thing. You only look at these 7 spots. The other thing is that sometimes you don't need to even sequence the 7 spots. What you do is you sequence, for example, 2 out of the 7. The reason is this. The haplotypes are such that these sequences at these spots are stereotypical. What that means is, let's say at this A, you see an A. At this B, T, G, C, A. Okay? So this is what your one haplotype. What that means is that if I look in this population and sequence these 5 sites, frequently I'm going to see, if I see an A at this site, I frequently will see a T at the second site. Okay? I'm not going to see a G or a C or etc. Right? And if there is a, for example, if I have a C at this site, let's say A, C, T, C is another haplotype. Okay? So if I have a C at this site, the frequency of me seeing an A at this site is very high. The reason for that is since these travel like a block, what that means is these 5 locations, they won't have every combinations of ATGC. They are going to have a limited combination of these sequences. Does that make sense? So if it was every combination, it would be 2 to the 5 or whatever, right? So that many different sequences we can see. Instead of that, in this population at this loci, I'm going to see 2 or 3 different haplotypes. Okay? So instead of then sequencing even the whole 5, I can say which of these SNPs distinguish these different haplotypes, then I can even sequence only those 2, for example, right? And that will reduce the cost of GWAS a lot. And this is very essential because the power of the GWAS comes from looking at many, many, many individuals. Because if you don't do that, then you can't correlate a certain SNP. But that also means, let's say, in this example, only 2 of these I'm sequencing only these 2 SNPs, right? Because they tell me all the information that I'm going to get from the different haplotypes. Then I'm going to be saying, okay, these SNPs are linked to a disease, right? But it's not telling you what mutation or what sequence in this whole region is linked to the disease or causal to the disease. It's just telling me something around the region of these SNPs are linked to the disease. Does that clarify? Very good question. I'm glad you asked. Any other questions about this? All right. Okay, so the first thing is done. We need to know human variation. We need to determine all the different haplotypes, right? Because a lot of the GWAS studies depend on the haplotypes. But for example, one of the things that you might actually recognize from this table, which summarizes the ethnicity of the participants in genome-wide association studies, you can see that the number of European individuals has been really millions, right? But not so many from other parts of the world. What the limitation of this is then, what you will know is you'll know all the haplotypes that are within the European populations, but you don't know the haplotypes from the rest of the world. So you actually won't know what the normal variation of the sequence in the genome is. Okay, so it won't. Then, if you see a SNP and then you go to the region and you say, I have this gene X, right, and all of the European individuals have exactly the same sequence of this gene X, except, let's say, so you found that your SNP is associating, you went to this region and you saw your gene X, right? And then you looked at all these individuals in your population, and you say, okay, there is one site here which is an A in all of the disease individuals, and in all of the normal individuals, this is a T. And you say, okay, here's a really great starting point, because I know that this A is in the gene X, and I see it in the disease. Is this presence of A in gene X causing the disease, right? That's a great starting point, if that's the information you have. But let's say that in other places in the world, there are plenty of people with the T sequence at this site, and they're all, plenty of people with the sequence of A at the sites, and they're all diseased, right? If you knew this, then you wouldn't have hypothesized that this sequence is actually causing the disease or not, right? So to understand GWAS and link it to the disease, then we really need to know actually what the normal variation is. Yeah?
Is the disease for A, wouldn't that support the hypothesis?
What do you mean? Okay, let me give the example. Okay, so this is disease is A, right? And the normal is, let's say, T. And you're saying, is this A causing the disease, right? But if you look at other populations, you'll have lots of normal people with A's, right? And then your hypothesis is not correct, right? So the limited information is going to restrict the accuracy of your hypothesis. All right. Some of the things that we learned from GWAS is that GWAS is great for diseases and phenotypes that are caused by one gene. Okay, it's really easy to find them, and Human Genome Project and sequencing and the GWAS really enabled us to pinpoint these genes. What are these? One gene, one disease type of situations. But for a lot of other type of diseases are shown here, the number of loci that are associated with the disease is very high. What that means is the number of genes that contribute to that disease is very high. Okay, so these we call the complex diseases. The disease is result of many, many genes mutations. And it's difficult to then find which of these genes are actually contributing to the mutations, because their contribution might be very small, okay? So, and that's why it's very difficult now to actually find the genotypic basis of complex diseases, and that's one of the biggest, for example, goals of National Institute of Health, to figure out how to solve this problem. Because, for example, you can carry a certain SNP sequence that's associated with a complex disease, okay? Let's say you went to one of these DNA testing companies, you got your sequence genome, they looked at a bunch of SNPs in your genome, and they said, okay, one of the SNPs is associated with heart attack, right? Don't get a heart attack, because heart attack basically is a very complex disease. Many, many genes and SNPs are associated with it. What that means is, you may carry a certain SNP sequence that's associated with the disease, but you're almost equally likely to have or not have that disease, okay? Because that SNP is statistically linked, but its contribution is so small that you can't conclude whether having that SNP is going to give you that disease or not, okay? So but that's for complex diseases. Of course, there are some SNPs that are clearly associated with disease like BRCA1, etc. Right? So I'm talking about complex diseases like obesity, etc. And one of the complex phenotypes is height, for example. So there are lots of hundreds of genes that contribute to the height of an individual, but each of its contribution is so small that we can't say, okay, if I have this sequence at this gene, I'm going to be tall or I'm going to be short, etc. Right? So their explanation power is very low. All right, so that was one of the reasons, for example, a few years ago, FDA stopped this company called 23andMe selling genetic tests that are related to disease. The reason was that 23andMe or the researchers, it's very difficult to tell if a person has a SNP is going to have that disease or not, right? So basically this genetics company, when it's founded, essentially said, okay, you have a 20% chance of getting a heart attack, right? And that's not very good because it's not accurate. So the FDA said, wait a minute, you actually can't say that, right? So it said that these inaccurate test results could cause people to undergo unnecessary health procedures because people actually don't know what a GWAS is and how to interpret that, right? So 23andMe stopped selling this for a while. And then in 2014, the company basically still sells the personal genome test. That's the ancestry type of test. It'll tell you what percentage Neanderthal you are and stuff like that, right? And what region of the world your ancestors come from, etc. But then they suspend all the health-related results. So in 2005, the company actually cleaned up some of its practices, and the explanation of what these SNPs and their interpretations are. So in 2005, now it's allowed to include more reliable health information for about 36 diseases. So these include cystic fibrosis, sickle cell anemia, and Tay-Sachs, etc. Right? So if you get a 23andMe health panel, you should read their explanations of each of these, actually what each of these means. So this type of GWAS is really great to go after disease, etc. And there are also some stupid uses of this, right? So you can always come up, if there's a technology, we'll find a way to use it in very stupid ways, right? So for example, in this example, is that the person is going to take the DNA of this poor little child and then see if this child is going to be great at sports or not, right? So is that really necessary? Probably not, right? And in the article, actually, it says, Dr. Foster suggested another way to determine if a child will be good at sprint and power sports. Just line them up with their classmates for a race and see which ones are the fastest. So, okay, I'll see you guys tomorrow. Remember, tomorrow is there's class.